Hi, folks. So I got a really interesting email from a student who was asking me to kind of clarify a little bit about um, the Avery, McLeod, and McCarty experiment and kind of help them understand a little bit more about what was going on, what the different states were for the bacteria that were involved, and how genotypes and phenotypes were connected. I know with the Avery, McCarty and McLeod experiment that there are kind of a lot of moving parts. So I thought I would do a quick explainer video and see if this was helpful to you guys. Okay, so how does transformation change rough bacteria to smooth? What's the situation that we're seeing? What do we mean by rough and smooth? This is a very primitive Dr. Pickett's normal animation quality, but I hope it's helpful. Okay, so let's start out with a smooth bacteria that has the normal capsule genes present. So the genes that are required to produce the bacterial capsule are wild type. And when those capsule genes are expressed, they're transcribed and translated, what's going to happen to that bacteria? What will its phenotype be? Well, slowly, individual sugars will be polymerized together into long polysaccharide chains, and the bacteria will end up being surrounded by this kind of jelly cloud of polysaccharides. So now you literally can see there is a physical barrier between the outside culture media or between the blood system or the peritoneal, the tissue fluid of a mouse, and the bacterial cell wall, kind of shown here in orange. Here in red is the bacterial circular chromosome. Capsule plus is the wild type capsule gene that's present. Now, when we're boiling these capsule plus cells, these smooth cells, what's going to happen? Well, as we increase the temperature, eventually the polysaccharide coat is going to be solubilized in solution. The cell wall is going to be popped. The DNA, the bacterial DNA chromosome is going to go into solution. But you'll also see there's going to be some shearing of that DNA. So little fragments of the bacterial chromosome will be produced, including fragments that have, for instance, the genes required for capsule formation. So here I'm showing this little arc of DNA there in dark red, maroon as we like to say at Loyola, and it contains information on it for the wild type capsule gene. So what's happening in transformation? We've popped these capsule plus DNA containing cells. We've added that smooth juice to rough bacteria. What does that actually look like? What is the process of transformation? Well, let's set up a system where we've got some boiled smooth juice and capsule plus containing DNA in solution, and we put a rough bacteria, capsule minus bacteria, into the solution with it. We can see the actual transformation event. So here we've got our capsule minus cell. It's got a mutant form of these polysaccharide synthesizing genes on it. It does not produce a polysaccharide coat. You'll see here, there's no polysaccharides surrounding the bacterial cell wall. But because we placed this cell into boiled smooth juice, where there are little fragments of the capsule plus chromosome present, this DNA now is available to physically interact with proteins. We used to call these proteins the activator proteins. They're kind of sitting on the cell surface, and they're kind of responsible for reeling in that free DNA that's present in solution. So what's going to happen? Well, you bet the capsule plus DNA is going to move across the cell wall. It will go from being out here in solution to being within the cell proper. Then our old friends, the recombinases, are going to find regions of similarity between our capsule plus containing DNA and our capsule minus gene, and they will initiate a recombination event. Capsule plus will move out of trans to the bacterial chromosome that has capsule minus present by having that bacterial chromosome move from being in trans to capsule plus to in cis through recombination. Remember, with recombination, we have to cross every bridge both ways when we encounter them. So the bacterial chromosome will go up and capture the capsule plus gene, then recombine, moving capsule plus from trans into cis. Similarly, capsule minus will be captured by the fragment. It will move from being in cis with the bacterial chromosome to being in trans to it. Once capsule minus moves off onto that little fragment, that piece of DNA will eventually be destroyed. So if we have a successful recombination event, what's going to occur? Well, our capsule minus gene will be, through recombination, converted into capsule plus. Those genes will begin to be transcribed, RNAs will be made, proteins will be translated from those transcribed RNAs. Those proteins are required for polymer synthesis to make a polysaccharide, and boom, these long sugar chains will be delivered to the outer surface of the bacteria, and a nice slimy capsule will form. So anyway, guys, I hope that is helpful. I hope that answers the email question that I got. I think that should help you guys visualize it a little better, but just let me know. Thank you very much for your attention. I noticed we had 100% completion on the exam today. So thank you very much for doing your exam. And also have a great weekend. I will get grades out as soon as I possibly can. Have a good day, my friends. It's Friday evening, but anyway, Dr. Pickett is thinking of you guys and hoping that this is helpful. Take care. Bye-bye.